Started, I'll introduce you here. So, let me hit record. Or do you? Oh, you already said it, huh, Inez?
Yep.
Looks good. Okay, good. All right, everybody. So it's my pleasure to introduce today's speaker, and I'll try not to butcher your name. Kyle Biegasiewicz. Is that close enough? Or
That's good. That's good.
pretty good? Okay. Yeah, you can correct me. Nobody ever gets my name right, so I'm always sensitive to that, and I butcher others. So anyway, so I had the pleasure of working with Kyle this last year on this large multi-institutional grant, um, and just one, I still owe him a ton because he really carried us across the finish line with this thing, and I was just really extremely impressed by his research directions, and he was just a tremendous colleague to work with. And I can't wait to work with them in the future. Whether that one gets funded or not, let's fingers crossed. We haven't been rejected yet, so that's always good. But either way, I think we're going to find a lot of paths to work together because there's a lot of overlap. But I also thought, a lot of overlap with research going on, not just here, but as this is a joint colloquium now, in particular at NAU and also ASU. And as we were talking, I was able to, Trevor and Kyle are able to meet because ASU is such a huge community that sometimes it's easier to meet this way through things like this. So really happy to have him. So Kyle received his BS in chemistry from Niagara University in 2010, and then went on to University of Rochester to work with Bob Beckman, I believe, right? And got his PhD there in natural product synthesis and catalytic method development. Um, and then after completing his PhD, went off to Princeton to work with Todd Ister in designing photoenzymatic strategies and address challenges in radical mediated synthesis. This last, I guess it's a year and a couple months ago, he joined ASU, and what a tough time. I mean, so you've basically been there throughout all of COVID, I guess, at this time, and as a professor of chemistry with the School of Molecular Sciences, and his group there works on and also also part of Biodesign Institute, I believe, correct? So part of that amazing center there as well. And his works on the unique catalytic capabilities of these underexplored enzyme classes, some of which I really hadn't known a lot about till we started working on this proposal together. And I'm just fascinated by them and what they can do. So I think we'll hear some of that today. And the idea of rapid construction of complex molecule synthesis. So with that, I'll hand it over to Kyle. And again, thanks for joining us. Really looking forward to this talk.
Absolutely. Thank you so much, Gabe. I really appreciate that kind introduction. It was really fun working with you on some grants, and I'm seeing some very familiar faces in the crowd and some new faces also. So that's really great. Yeah, today what I thought I'd do is sort of give you all sort of an idea of sort of where I sort of come from, sort of in the research space when we're talking sort of about complex molecule synthesis. That's sort of what I've done all the way throughout through sort of my PhD, my postdoc, and currently in my group. And I'll give you some insight into sort of what I worked on sort of in PhD, and then sort of also what I worked on in Princeton, and then we'll talk a little bit about how that sort of influenced what I work on now here at Arizona State. And so before I get into sort of the I would say the meat and potatoes of the talk, I kind of wanted to go briefly just through sort of why exactly I care, and the group cares about sort of complex molecule synthesis in general, and sort of so how I figured I'd do that is kind of bring up a few sort of frequently asked questions about complex molecule synthesis. One of them that I typically get as sort of new students start to join the group, or I talk to sort of my fellow colleagues, is that is it still a worthwhile pursuit? Have we made all the molecules that we ever need to? And is it are we kind of just doing it for fun at this point? And what I would say is it definitely is, right, a worthwhile pursuit. And that sort of, if you look sort of historically at sort of the Nobel Prizes, roughly about 30 years or so, there ends up being sort of a new Nobel Prize in organic synthesis, and sort of anything from sort of the early work sort of on synthesis of purine to sort of the I guess these are sort of interesting ones, right? R.B. Woodward for organic synthesis, that's pretty unique. And then E.J. Corey in retrosynthesis, right, the backward technique, is this is obviously shown a very to be a very important area, right, in research. And I'd even say sort of kind of meeting the sort of every 30-year standard. Directed evolution, right, with Francis Arnold and others, just one where we can use directed evolution of proteins to synthesize molecules, and we'll talk a little bit about that a little later in the talk. But then another question I usually get, is, what does the funding landscape look like? What areas are sort of left for innovation? And sort of what I thought I'd do is maybe just present sort of a few different areas that are sort of the areas that are sort of keenly focused on at the moment. Right, so we have sort of a number of players that are interested sort of in sort of strategic bond construction, right, with known methods, right? So they're kind of the ones that kind of take everything that, that's developed sort of on the methodology end, and they can develop that into sort of these really nice, complex molecule syntheses. We have people that are sort of thinking about sort of metal-based methods that are focused on generation of sort of carbon-carbon bonds, carbon-oxygen bonds, or carbon-nitrogen bonds, for example. And we even have sort of Sergey Peronin here from University of California, Irvine, even asking, there's so many people, is it even cool anymore? Then we have sort of single electron chemistry, and we'll talk a little bit about this later on in the talk, but sort of using sort of light or electrochemistry to sort of initiate sort of these carbon-carbon bond formations, for example. And then what I would say that, over time has been sort of considered an emerging field, but now I would say it's a field that's sort of here to stay in sort of the organic synthesis arena, and that's in biocatalysis. So thinking about how to use biomolecules, oftentimes enzymes, right, to start to make organic molecules. Another sort of keen question, right, people come up with is, why are people interested in the subject? And I think this, sort of hasn't changed a ton, right from the beginning of sort of organic synthesis in general, right? And you have sort of biological relevance, right? So we're maybe constructing a molecule for sort of antibacterial, anti-cancer type of activity, for example, or for another purpose that you can imagine the range that sort of exists there. Structure determination is still a very hot area in the area, and sort of in that we'll construct molecules based on sort of a proposed structure of, for example, a natural product, to prove that structure is indeed the natural product, right? So you isolate it from a given source, and then synthesize the compound and compare. But I'd say the area that I'm most focused on is sort of this area of organic synthesis, and that I often think of complex molecules as, right, the ultimate platform for new reaction development. For example, if you can perform a reaction on benzene, right, you're often going to wonder, okay, if you have sort of that aromatic compound or aromatic moiety on a compound, can you still sort of do that sort of transformation on a pretty complex molecule? And I think one good example sort of is from Tom Mimoni's group at UC Berkeley, right, where they've been able to sort of develop these radical mediated transformations to construct pretty complex natural products. But you start to see sort of this, one of the things that they realize in sort of doing this is that they need to screen up to sort of these 30 chiral files in order to kind of have selectivity here, right? So this worked really well, right, on a smaller molecule, but as you start to look at it on a more complex molecule, it gets a little more complicated. And I would say this is more maybe relevant for the students that might join the group. Oftentimes, I'm almost asked this every time, for people that do complex molecule synthesis, don't they always kind of look like this? Sort of like, it's always taken sort of had sort of reputation of being sort of these very angry and competitive individuals in the lab. I would say that we've sort of replaced that sort of anger and competitiveness with a little bit more sort of innovation and kindness over time. And sort of we've been more welcoming, right, to sort of the diverse type of approaches that we take to complex molecule synthesis. And so maybe on more of a serious note, sort of, if you follow sort of the current trends in drug discovery, there's sort of a constant increase in target complexity, right? So if you think about sort of the average time for from going from lab bench to drug is somewhere around 15 years. This number really hasn't changed over a long period of time. And the reason for that, one of the many reasons, is sort of the complexity of drug candidate is rapidly increasing. So we're looking at somewhere around 20 steps is sort of the average synthetic pathway, right? So that's pretty long still, I would argue. And if you're thinking back to sort of your organic chemistry days, your sophomore organic chemistry days, where you devise maybe a small retrosynthesis, and that would be sort of a small molecule. This is sort of what small molecules look like nowadays, right? These two molecules are considered to be small. Sort of, for example, one example here is in Halovin, this anti-cancer agent. Right now, even in industry, right, they're taking 59 steps to synthesize this molecule. So you can imagine that's quite the endeavor to go through in order to access this compound. But they do it because they need it, but at the same time, right, they have a long way to go there. And then sort of in this case, right, we have sort of this to be sort of considered a small molecule. I would also argue is it's a pretty complex molecule, and not something that you can just sort of write down on paper real quickly. So I would say sort of the complexity falls in sort of not only the functional groups, right, that are on the molecule, but also in the stereocomplexity of these, right, as we start to go after specific targets, for example. And so what I wanted to do is kind of take you through sort of three general stories as sort of what motivations were for me and sort of going after sort of complex molecules in the past. And the first one actually started in the Beckman group when I was at University of Rochester. There was this individual that actually came to visit us from the Max Planck Institute of Molecular Physiology, Herbert Waldman. He gave this phenomenal talk in sort of complex molecule synthesis. And one of the things that he had discussed with us is sort of this molecule here in Rasmonin. This is something that the Beckman group had been involved with a while ago in sort of the total synthesis of it. But again, typically these total syntheses end in like 1 or 2 milligrams generated, and usually it's not enough to really sort of pursue, let's say, the mechanism of action of a compound. And so this is sort of a quote that he shared with me. He said basically that this molecule, right, has the potential to be a real game changer in cancer therapeutics, at least as a lead. And sort of the only problem is across sort of 3 collaborations, right? They've only been able to work with 2.8 milligrams total. That number was very small to me, and sort of as a very eager young faculty member over some Rochester beer and barbecue, I decided, you know what, I want to be the person that gets them to sort of this dream gram of the compound. I of course signed up for this without my advisor knowing, and so we had I had a stern talking to, but he said, you better get it done now. So we went ahead and pursued that. Just to give you a little bit of background about Rasmonin, it was actually isolated in 2000 from a couple of different species and groups. Its biological activity features sort of this demonstration of selective destruction of RAS-dependent cells with an IC50 of 0.16 micrograms per mil. And it can present a novel class of cancer chemotherapy, therapies in that it's a sort of treatment of tumors expressing, in particular, active mutant RAS. And so the goals in this for us were to develop a synthesis of Rasmonin that would be amenable to scale up and synthesis of analogs, as well as improve the understanding of the biological mechanism of action in collaboration with the Waldman group. And so just to take you into the insight, so if you look at sort of the molecules that I showed you from before, and you look at this one, right, this one's not as complex, I would argue, as sort of the ones that I showed. But sort of what made this one particularly challenging is actually the installation of this group, sort of highlighted in blue in the top right. And that's a hydroxyl methyl group. So essentially the chiral installation of methanol, right, onto a molecule. And so just to take you through sort of what a scheme would look like to install this, typically, and this was how we did it in 2006, some colleagues before I arrived to the group, right? You'd start with sort of the installation of this chiral auxiliary in this first step here. You then take this through a titanium-mediated sort of enolate formation in addition into what's a protected form of formaldehyde, right, and bomb chloride. And then you go through sort of a series of manipulations, and ultimately, right, you have to take off the chiral auxiliary, and then eventually you get to sort of the simple aldehyde in the bottom right. And what I would say is sort of this is a ton, right, to do to get to just sort of a small molecule in the bottom right. And so just to highlight some of the thoughts we had going into what I'm about to tell you, right? So this stoichiometric auxiliary installation is not ideal You also have to take it off later on. And then if you look specifically at the step where you actually install the hydroxymethyl group, where you have air and moisture sensitive reagents used, titanium chloride, and then bomb chloride, that has a molecular weight of 156, right, versus 31. So you're looking at about, some pretty rough sort of atom economy when it comes to that, right? So what we drew up on the page is, okay, the holy grail then would be able to sort of take a simple aldehyde on the left, be able to use a catalyst and a formaldehyde source, convenient formaldehyde source, and be able to go ahead and do this hydroxymethylation stereoselectively and on scale. So I'm not going to take you through sort of the development of the protocol that we developed, but what I would say is, just to summarize, we developed in sort of collaboration with Novartis the hydroxymethylation of simple aldehydes, where you take sort of the simple aldehyde, put it in the presence of formalin, so it's aqueous formaldehyde, right? Otherwise, you'd have to use it in gas form. That wasn't ideal for us. And in the presence of the Jorgensen-Hayashi catalysts here, we can perform just some enamine chemistry, and we can sort of add to formaldehyde. Another sort of equivalent of formaldehyde adds this, and we end up getting to this lactol intermediate 1. And then what we can do is take sort of what I would say is sort of this, sort of acid sensitive intermediate, and take it through either pinnic oxidation or vitigolithination to get to some isolable compounds. And sort of these are sort of the parameters that we optimize sort of around the horn here. Right, we need fresh purification of the aldehyde is pretty important. And then sort of we played a lot with the buffer composition, as long as as we sort of scale this process up. And if you have any sort of questions about this, feel free to ask towards the end, or put it in the chat. Yeah, so the reason why we actually initiated this process sort of with Novartis is actually this actually played a role sort of in one of their drug development campaigns at Tecturna. And what we found, what ended up happening is, sort of in the middle of the collaboration, they were actually giving us this Jorgensen-Hayashi catalyst, and the compound failed in phase 3. So that stopped our collaboration. But we wanted to keep going. We wanted to be able to do this on scale, right? So we had to think about we had another situation where we have to come up with a way to synthesize this thing on scale. And so we got to doing that. We had a bit of time that we worked on this, where we took sort of proline. We would go ahead and do a Bach protection, do methyl iodide treatment to do the esterification in the top right, and then treat that with 2 equivalents of Grignard reagent, followed by sort of sodium hydroxide treatment and TMS protection. We can go ahead and access this compound in sort of on a 100-gram scale in 1 pass. There was a lot of good sort of large scale up. It was a whole lot of fun. We ended up finding that this protocol was sort of relatively easy enough for undergraduates to do. So what we did is we turned this into sort of a real world type of application for sort of undergraduate students to generate this Jorgensen-Hayashi catalyst. They work on it on about a gram scale or so, and so for every batch, they produce, I think there's like 300 or 400 students in that, right? So they produce about 400 grams a year of that, and they get to use it in sort of new types of reactions. But that's a paper we sort of just submitted this year. But back to sort of the task at hand, right? We want to be able to make Rasmonin. So the retrosynthetic analysis that we did, or affiliated with this, is we sort of take Rasmonin, be able to split it up into sort of a nice convergent synthesis via Yamaguchi esterification between this alcohol here and this carboxylic acid. Those two would then come from this Mukayama Aldol DBU rearrangement and this Horner Wadsworth Emmons olefination, which we'll be talking about shortly. And then what we wanted in this case is to have not only convergence in sort of the molecules, the size of molecules coming through to eventually produce Rasmonin, but we also wanted to create sort of a convergent strategy in terms of the protocol that we get to sort of achieve sort of our stereoselectivity. And so what we thought is we might be able to use this hydroxymethylation vitigolithination that we developed to start from these simple aldehydes 6 and 7. So to take you through sort of the forward route for this, right? We start with propionaldehyde. We go ahead and do the hydroxymethylation and the vitigolithination We produce 10 grams of this intermediate 8. We then do a hydrogenation, a stereoselective hydrogenation using this rhodium NBD catalyst. We then exchange this hydroxyl group for the iodide. Then we do a reductive coupling to get to compound 11. And then finally, dye-ball treatment gets us to 7 grams of this in one pass. This was actually really challenging also to produce on scale before, using sort of an auxiliary-based method also, where the most we ever had in hand before we started working on this was, I think, 50 milligrams. So that got to be really challenging, from this end too. So to be able to produce 7 grams of that in one pass was really exciting. We then take that aldehyde and we put it through sort of this vinylogous Mucam aldol type reaction with furan seen here, and it generates this butenolide in 12. And then at that point we hit it with dye-ball. We do sort of reduction of this lactone. We can then initiate a DBU rearrangement from the 5-membered to the 6-membered ring. And then manganese oxide oxidizes what would be this corresponding lactol to the corresponding lactone. And we can access 2 grams of that in one pass. If anyone's at all interested in sort of talking about sort of the transition state of this, I'd be happy to talk about it sort of after the talk. For the southern hemisphere component, so what we did is we went ahead and TBS protected, right, it's commercially available, very cheap, right, diol. After sworn oxidation, we get to compound 7, or simple aldehyde that we propose using. Then we take it through our hydroxymethylation phenol protocol again, right? 20 grams pass in one pass, really, really good. And then after a dye-ball reduction and Park-Doring oxidation, we access compound 16. That's then followed by a Horner-Wadsworth-Emmons reaction, then lithium hydroxide treatment to get the corresponding carboxylic acid here on a 10-gram scale. So to sort of wrap up the synthesis, we went ahead and subject these compounds to Yamaguchi conditions up top. And then after HF treatment, we can go ahead and access corresponding diol and ultimately Rasmonin, right? So we sort of doubled the goal so far. We just published this, I think, 2018. And we were able to achieve, a whole bunch for Herbert Waldman, who's now working on the mechanism of action. We just checked in, we just had a meeting, and I think they're still working on it. So it's pretty interesting. He actually didn't get the compound, I think, till the start of 2019. But at this point, right, I actually had no real reason actually I really wanted to continue in sort of synthesis, very similar to this one. Right? I love building big molecules. Thought it was really, really cool, and really had no intention of using enzymes at all, in my life for organic synthesis. But I actually ran into Todd Heister at Princeton at a Gordon Research Conference, and he sort of encouraged me to join his group as a biolec postdoc. And so what they were interested in is sort of this sort of bio-inspired light escalated chemistry, where we can use, we wanted to address the scientific challenge of precisely harnessing the power of light for making and breaking chemical bonds. This is one we got some funding with, funding from the U.S. Department of Energy, and sort of, we had a ton of partners sort of in industry as well as across a number of different academic institutions as well. That's actually where I got to meet some ASU colleagues for the first time. It was very, very exciting. Just to give you sort of a map of what this looks like. It's a little fuzzy. We still haven't had one that's not fuzzy, but ultimately this is sort of the layout. I'm sure many of you know Tom and Anna. And sort of, we had this sort of huge cohort. It was largely based in Princeton. We've worked a lot in sort of photoredox catalysis in the past, but sort of very exciting time for us. But before I sort of get into what I'll tell you about sort of in the enzyme arena, I wanted to just give a brief introduction and sort of how I guess my group as well as Todd's group thinks about using biocatalysts in complex molecule synthesis. And so we kind of sit using this the statement up here, and that complex molecules inspire, right, the development of biocatalytic technologies. One of the most inspiring sort of carbon-carbon bond formations in across organic chemistry is this conversion of squalene to hopene using squalene hopene cyclase, an enzyme that's found in nature. And then the idea would be sort of to have this sort of second phase where after you've gone through the scaffold construction, be able to start to perform selective elaborations of that to access new chemical space. I should say that organic chemists have been working on trying to mimic this exact step for quite some time. And I think as far as we know, we've been unsuccessful in doing so. So that's been really interesting to me for a long time. And so what we believe, right, is that enzymes can provide selectivity across all phases of chemical synthesis. And sort of one of those Nobel Prize winners that I mentioned before, he studied actually this reaction mechanism from squalene to hopene. And I don't need you to read this whole line here, but one of the more important things to mention is sort of this is being in 1959, right? He wraps this statement up by saying the human mind is not quick and clever as an enzyme. And I thought that was pretty inspiring in that sort of now we sort of think of this in sort of a way that we kind of respond and say, okay, this is definitely possible. Maybe we are quick and clever as an enzyme. I don't know if I'll ever be that, but it's important to note that this goes through sort of carbocations right up top, but there's a lot of other sort of reactive intermediates, right, that enzymes are able to sort of stabilize and utilize in from organic chemistry perspective. And right, that's anything from sort of carbocations to radicals, carbanions, etc., oxoniums, even homoniums, right? They can do halogenation type chemistry. And so, what we started to be inspired by is sort of exactly that directed evolution process that I mentioned from before. And sort of what that workflow looks like is that you start with a parent protein. You perform sort of mutagenesis, where you mutate the DNA. You access a series of new mutant proteins. You go ahead and create sort of a screen and selection phase for that. And you can screen about 1,000, right, over the course of 2 to 4 days. And then what's so great about this, right, is that you can go ahead and this whole process is iterative, right? So you can go ahead and repeat it to continuously evolve the protein. And so what you're doing, right, throughout that whole process is you're navigating the fitness landscape. So if you can imagine, right, you want to do a ketone reduction, for example, you can go ahead and sort of in this black space, right, it's not doing that ketone reduction, but sort of on these mountainous regions, right, that's where it actually does have some activity in that regard. Once you're able to access these, and oftentimes that's accessed strictly by sort of taking a peek at a parent or a wild type. You can go ahead and start to navigate that hill via evolution, right? Via the same kind of protocol that's up in the top right. And so from our perspective, the process kind of looks like this, right? You have some starting enzymes, right, that have sort of native function, but might have sort of the mechanistic capabilities of possibly performing the reaction you're hoping to perform. Through an interrogation process where you clone, express, and assay them in whole cells or cell lysate, you hope to identify a catalyst that has sort of an overlap between that native and new function. And so when you start to access sort of this space sort of in purple, right, you've now achieved what's sort of considered promiscuous function. So before, people were very interested in being able to do sort of natural reactions with substrate promiscuity, right? So they can accept new substrates to do very similar types of reactions. Now we're looking at reaction promiscuity, right? Where we can actually turn on new function for the enzyme. And then ultimately what you can do is you can take that identified catalyst and you can evolve it towards the way that you want, right? So you can climb sort of that new function hill. And sort of the goals that we have is we want to solve a synthetic challenge, right? So if this challenge that we're going to go after has been solved from a small molecule perspective, it's not going to be something we're going to pursue. We want to get after something that really only the enzyme can solve right now. And then we obviously want something that's catalytically promiscuous. And then we have sort of a series of metrics that we turn to, and I just kind of jot them down the bottom right. So how can we train enzymes to perform new functions? This is sort of a question that we ask sort of all the time in my group, and we definitely did in Todd's group. Sort of the many different options kind of fall in sort of using the same mechanism to kind of turn on new reactivity. So kind of use the mechanistic machinery in the enzyme subjected to new substrates that might go through a similar type of mechanism and perform sort of new reactions. We can also string together, right, a series of enzymes to make sort of complex molecules. One area that we have going on in the group right now is putting enzymes under pressure for evolution. That's something that's definitely in the early go, but it looks like it's going to be very promising. Very kind of cool area there. One area that I'll be talking about right now is sort of subjecting the light, especially for enzymes that have a redox active cofactor. And then electricity is sort of another area that we're starting to dive into now. And so when you think about this, right, as I was talking about in the previous slide, right, if you want to think about sort of biocatalyst development, right, we always think of enzymes as sort of we take something from nature and they're sort of single mechanism catalysts and we sort of use them as biocatalysts. But it's weird because if you think about sort of small molecule catalysts and even just sort of the copper box catalyst, seen here, we think about this catalyst or this ligand being able to sort of initiate a series of different reactions, right? Just by subjection to a different substrate, right? So we're going to use sort of an analogous idea, right, as we go into pursuing new reactions with enzymes. And in the Heister group, we're interested in seeing if we can expand the mechanistic versatility of enzymes through radical mediated processes. So one of the core families of enzymes that I worked with when I was in Todd's group is with these ene reductases. Typically what they're known for is taking sort of these Michael acceptors, right? So an alkene with an electron withdrawing group to the corresponding saturated compound, right? So this is via hydride addition and protonation. And what we love about these enzymes, right, is that they are largely substrate promiscuous, right? So they're able to sort of across the family, they're able to tolerate all sorts of different types of structures, right? Even up to sort of the larger ones, for example, the steroid in the bottom right. The typical mechanism, right, that these go through, they toggle between the FMN oxidized or quinone form and the reduced form, the hydroquinone. And that reduction actually happens via cofactor and NADP or NADPH, I should say. And then this is then recycled via another enzyme. So ultimately, what you do is you have sort of reduction of the oxidized form. This delivers a hydride to your Michael acceptor, and then it continues going throughout that catalytic cycle. One of the things that we thought about heavily in the Heister group is, not only can these perform, two electron chemistry, but they also can perform single electron chemistry, right? So they can give up an electron and start to reduce substrates, right? So right here you have, loss of an electron and a proton, or addition of a proton. You end up at the FMN semiquinone here. And then it's just another reduction, right, to get you to the hydroquinone, right? So before we were toggling between this oxidized form and the hydroquinone, but now we have sort of this intermediate to take a look at. And in 2017, as I was sort of joining the group, some of my colleagues were successful in taking these alpha bromo esters and using just the ground state reactivity of the FMN oxidized form to go ahead and affect sort of the generation of the semiquinone here. And then ultimately, since it was in sort of the chiral pocket of the enzyme, you could start to perform selective hydrogen atom transfers to the radical generated here after homolysis of that carbon-bromine bond. Ultimately, what you end up accessing is sort of pretty decent yields, but then more importantly, very high enantioselectivities selectivities. This was at the time, right, the state of the art in terms of hydrogen atom transfer, selective hydrogen atom transfer, I should say. It's very challenging to access. But we had sort of a larger question, right? Those alpha bromo esters are pretty easy to reduce, right? They have pretty low reduction potentials, right? So we started thinking, how do we start to expand the redox window of flavin to access substrates that are more challenging to reduce? And the thought process was to go ahead and hit them with light, right? So it's known that, generally if you go ahead and oxidize, or sorry, if you photoexcite these intermediates, right, they have higher reduction potentials, right, and they can start to reduce challenging to reduce substrates, right? So what's pretty cool about this, and sort of the way we thought about it, is if you do the photo excitation, right, it's toggling actually between sort of all 6 of these, right? So you have in sort of one well, right, you have potentially 6 catalysts that can do sort of the reaction that you're hoping to. That was sort of a really fascinating idea to us at sort of the onset. One of the challenges that we wanted to address is sort of an example like seen here, right? You have this alpha chloroamide, and you perform sort of radical cyclization of it and be able to control not only this stereocenter, but stereocenters that were more distant from sort of that alpha carbon, right? So before we were able to control alpha stereocenters, but now we want to be able to get to sort of ones that are a little bit more distal. And so if you do this this actually provides really good insight, and sort of if you do these reactions sort of in an achiral sense, you treat this sort of with an iridium photocatalyst under blue light irradiation and a sacrificial amine, and you generate sort of a combination of the cyclized product and what would be the hydrodehalogenated product, sort of just the radical generated here, and then hydrogen atom transfer occurs. And you generate a 73% yield, but you get a 1-to-3 ratio of the cyclized to reduced. And that was about as good as it got before we started using these. And that was not ideal for more synthetic chemists. So the process that we thought about is maybe we can go ahead and using reductases to do a photo-induced dehalogenation, get to the alpha radical here, right? And then we perform the stereoselective cyclization. This will bite down onto this alkene, and we'll end up with this radical out here. And then we could have a selective hydrogen atom transfer to sort of a more distal position. And so general scheme and scope kind of for this reaction, the reaction worked remarkably well. We actually found this mutant T36A of gluer that was able to affect this transformation on a ton of different substrates. That's usually not remarkably common, but we did luck out sort of in this one. What's really cool is that you can not only access compounds that are sort of have sort of the styrenal component to it with sort of this aromatic species, but we can also do this on ones with alkyl substituents. Those ones actually had actually not been available to us sort of in the literature at all. So we're very excited about that. We also have examples where we're able to sort of set that stereocenter in sort of distant stereocenter, but I'll be talking about that shortly. And the way we envision this happening, right, is that we'd start with sort of reduced FMN and the substrate in the active site. We'd have a photo-induced electron transfer, get to this alpha radical seen here. That would then go through a radical cyclization, get to the radical down here, and then selective hydrogen atom transfer from FMN hydroquinone will get us to our desired compound in FMN, and then the process would turn over after flavin reduction. As we got to doing this, sort of a lot of the more common wavelengths for photoredox catalysts are usually sort of in the blue, or sort of right in the green area, or red. But what we found actually when we did a sort of a light screen is that we had sort of this peak sort of right around 490 nanometers, which we refer to as sort of cyan light. And so that was sort of curious to us, in that sort of flavin absorbs in the blue. It didn't make a whole lot of sense as to why cyan really made this thing go. And sort of what we started to do is we initiated some UV Vis experiments. And what we found is sort of, right here, right, we have reduced flavin in the black line here. When we reduce flavin and then add the substrate, we end up having this sort of new peak form right around 490. And we believe that was a charge transfer complex between reduced flavin and the substrate. What we found, we wanted to make sure that something was consistent with that. So what we went ahead and washed that out sort of with sodium benzoate, a common binder for these enzymes, and we can wash that out completely. And so what this told us, right, is that the first step of the process, right, it's not diffusion limited, and it occurs sort of directly upon light absorption. right? So that complex absorbs the light and then initiates the process. Very, very interesting stuff there. We also did some ultra fast spectroscopy in collaboration with the schools group to get some time constants on a lot of these, and that was a heck of an exploration for an organic chemist. Obviously, I could talk a little bit about that later if you're interested. But yeah, it took me in the direction of being able to work with them. It was really fun. But ultimately, sort of the mechanism that we came up with ultimately is, you hit this 500 nanometer light, you get the reductive single electron transfer, 10 picoseconds, and then you generate sort of these radicals here, the semiquinone and this alpha radical here. You then perform CC bond formation to generate the radical in sort of that distal site. And then you get selective hydrogen atom transfer, 250 picoseconds there. And then ultimately, right, it turns over and generates your desired compound. Went ahead and scaled this up. We're constantly building sort of photoreactors in the lab. We built this thing to do sort of our gram scale setup. Very, very exciting. This thing works on a gram scale, 92%, 9460 ER, very consistent with what we saw on sort of smaller scale. For engineering purposes, we're always interested to know if we can do sort of this reductive chemistry, but open to air. Usually you have to keep this under sort of reducing conditions. But what we found is that the process is actually robust enough that we can go ahead and do this open air. So we can do this with sort of the normal turnover system as well as with sodium dithionate. Very exciting study. Had a great time with this. Got to publish in Science in 2019. I wanted to briefly touch upon one other project that we were involved with, published in 2020, where we actually, we did that fully reductive process in the last example, but we were interested in sort of being able to do this, so these redox neutral type of examples in sort of very similar type of conditions. We're able to perform sort of redox neutral type of cyclization of these oxindoles. And so just to briefly talk about that mechanism, it actually does not go through sort of that charge transfer process, and there's sort of a series of experiments that sort of support this. But what we ultimately use is sort of FMN, and we use trypsin, sacrificial amine, to access the semiquinone, and then that semiquinone being just a little bit more reducing than the hydroquinone, actually is able to perform that single electron transfer. And then you get to FMN, you have the cyclization here, and then a single electron transfer and oxidation actually can occur to access these oxindoles. That was a really fun one going on sort of at the same time. We were sort of in a nice little race to the finish. So it was a good time. But so there was still a remaining challenge, right? We need to be able to do these reactions, but we also want to be able to do them not just in an intramolecular sense, but in an intermolecular sense. If you think about the enzymes that are capable of doing intermolecular reactions, right? They're usually something kind of creates sort of an intermediate, very similar to, sort of the earlier work I was talking about there. Right? They go through sort of an enamine intermediate, right? Usually from condensation with lysine residue. And then ultimately you have sort of the cross-coupled product is essentially sort of an enamine catalyzed aldol. But being able to do this sort of with a transient radical species is much more challenging, right? You have this highly reactive short lifetime species, and you have this, again, competitive hydrodehalogenation to the left, and then alternatively, right, you have this hydroalkylation to the right, in sort of your desired process. And so just to make sort of a longer story short, I just wanted to show you we can do this. We're able to actually conduct this reaction in an intermolecular sense. And we just recently put this one out. We're actually, again, with cyan light with the ene reductase. Again, this Gluer T36A has been really good at being able to sort of initiate this reaction. And what we're able to do is ultimately take sort of the radical that's generated here, it adds across this alkene, and then we have selective hydrogen atom transfer in sort of a distal position, very similar to the previous project. Very, very cool results. So originally, right, we thought this went through the same exact type of mechanism where you have sort of the formation of the charge transfer complex. It sort of generates the radical and it proceeds. But what was pretty cool is we've done some further investigating, And in this case, you not only need to add in sort of this chloroamide here, but you also need to add in the styrene, right? So now we're talking about sort of higher order enzyme complexes, right, that are necessary for this electron transfer to occur. They're still working on sort of some more mechanistic details about this, but ultimately what you end up having is sort of this enzyme templated ternary charge transfer complex in order to initiate this transformation. And it indicates, right, the enzyme templates the formation of a ternary CT complex, which absorbs the light to facilitate the radical formation. Really, really fascinating stuff to us, and sort of that's sort of the area that they're diving into now in the Heister group. Just to give you sort of a sense of what it can do, right? This thing can go ahead and access a number of different compounds and sort of styrenol types of substituents. It tolerates a number of substituents, so the methoxys, the bromides, the CF3 group, etc. But we're not only able to set sort of carbon stereocenters here, right? We can set sort of these carbon oxygen stereocenters as sort of disguised alcohols, if you will. We can start to do this sort of even on cyclic structures as well. So very exciting results there. This just came out this year. I'm very excited about this one. So we're on to my independent career. So right now, right, as Gabe was talking about before, been here for a little over a year, right? Started during a pandemic. Very interesting experience for me. We are the devils here over at ASU. And I'm affiliated with not only the School of Molecular Sciences, but also the Biodesign Institute, and in particular, the Research Center, Molecular Design and Biomimetics, where we think a little bit about using DNA or RNA also as catalysts under sort of our biocatalytic suite of options. We are located in the Biodesign C, second floor, and everyone I talked to definitely thinks we're an enzyme lab. We are primarily an organic synthesis lab. So we do work, traditional organic synthesis, anywhere from that to sort of biocatalysis. The group has grown pretty quickly. We now have 4 master's students, have postdocs starting soon. We have 7 undergraduates, very, very hardworking individuals. Like I have a ton of people to thank, over time here. But very similar to what you sort of saw before, right? We're interested in sort of complex molecular architectures, right? And we really want to see sort of their translational potential, right? In terms of sort of, methods development, develop medicinal chemistry tools, and sort of go after biological targets. And we think about doing that, right, in sort of a scaffold construction phase, very similar to what you saw before. And also we think about sort of late stage functionalization as well. And so the 2 areas that we work in to develop catalysts, right, are these bio-inspired catalyst mimics, and then also actually using biocatalysts. So not just enzymes, but we really want to dive into some DNA, RNA, etc. And just to give a couple highlights to what we've discovered so far, I was very interested in sort of creating a solution, right, to this terpene cyclase problem. Usually when you use terpene cyclases, these are membrane-bound proteins, real challenging to work with typically. And usually what they do is they go through this little cation, and eventually that cyclizes into a whole host of products that are really difficult to control. What I would also say is that they also have sort of stereocenters affiliated with them, not drawn here, but that complicates things even further. And so one of the thought processes was to take, so easier to use enzymes right now, commercially available ones in the lipases that typically do this sort of ester hydrolysis. And that being their native function, right? They use sort of the catalytic triad to go ahead and hydrolyze esters, for example. And we wanted to use that, we wanted to tailor it a little bit to starting to generate allylic carbocations, right? So the idea would be to take sort of allyl halide that would coordinate to the halide and sort of initiate sort of a departure. You'd end up with a allylic carbocation. And hopefully what we could have is sort of identify a lipase that could lead us to sort of each one of these compounds. And so, these reactions are run in about the simplest possible way that you could, in that we just take sort of the lipase and hexanes, these ones primarily are immobilized, 1 mole%, and go after sort of a cyclized product. And what we find so far, we've actually accessed several different lipases that actually do these reactions pretty selectively. And there's been no evolution quite yet. So this is actually pretty phenomenal so far. Where we can access beta pinene, alpha pinene, limonene. We have one that does sort of the halide transfer. So the chloride leaves and then comes back, but it's on the carbocation. And then we can also access the allylic alcohols. This has been a really exciting project. This is actually led by an undergraduate in the lab, Pratika Gupta. Fantastic stuff here. And the other area I was, one day I was in the lab and I was really interested in sort of just like, can we just put together a molecule like really rapidly by using the oxyanion hole of lipases? And actually, one of my graduate students just grabbed all the starting materials and went in and tried sort of very similar to the previous one. We wanted to go ahead and access sort of the THC core. And so the process where we envisioned is that we could take sort of fluoroglucanol, we could add it to this aldehyde, and we can access this alcohol here. That would then go through an elimination, generate the carbocation, perform recyclization, and ultimately get to this compound. In the first try, we ended up with a 96% yield, greater than 21 DR. I couldn't have asked for more on that one. We've since been able to go ahead and access ones that sort of can be further functionalized via cross coupling. Right, so we access the chloride, the bromide, the iodide, and the fluoride. We're not only interested right now, there's many different projects going on, but I wanted to sort of highlight that we're not just interested in sort of strictly the sort of those scientific endeavors, but we're also interested sort of in the education space. So I teach medicinal chemistry here at ASU. And one of the things that we started to do is started to use PyMol to kind of introduce students to metabolic biocatalysis, sort of in the metabolism phase of pharmacokinetics. So we have sort of a software familiarization phase, enzyme substrate binding visualization, comparative drug binding, and then finally hypotheses that sort of lead them to sort of designing drugs. And this has been really successful. They've really enjoyed it. A lot of students feel they really have discovered the drug of the future, but should be really interesting to explore as we continue along. Finally, I'm part of a two-body move to ASU. This is my wife, Laura, seen here, and we've sort of initiated a whole bunch of efforts, sort of at least discussions, right, affiliated with sort of many of the sort of diversity, equity, and inclusion kind of topics that are sort of on the table right now, especially in the STEM fields. We just published this perspective alongside of a number of different sort of wonderful colleagues from different universities, large and small, including sort of PUIs, as well as sort of R1 type institutions. And this is maybe one of the more impactful papers I think I'll maybe ever put out with some people. Definitely, if you're interested in sort of our efforts in this area, I'd be happy to talk about those as well. But it's called Organic Chemistry, a retrosynthetic approach to a diverse field. It's out. I think it just appeared on our SMS website. So if anyone's interested, feel free to take a look. Just quick acknowledgements, right? A ton of people from the Beckman group. When I was at Rochester, this is Bob in the lab taking a look at maybe what I've done. And then this is sort of my co-captain on a lot of projects, Doug Tush. Fantastic. Obviously, a ton of the funding sources, really great. Over in sort of the BioLect crew at Princeton, right, we have sort of, this is the core crew that worked on sort of a lot of that enzyme mediated transformation stuff. The Department of Energy, very supportive in this process, and sort of a ton of funding also. And this is my group. We're ever growing. We started to do sort of the outdoor arrow pushing sessions. This is us welcoming our new undergraduate Daniela to the group, having a great time out there. We do this, and then we also, we headed out to Topgolf sort of at the end of last semester, having a good time out there. If anyone's never been to Topgolf, you're more than welcome to join us anytime. Looking forward to having you guys down, as soon as this pandemic sort of winds down. And with that, thank you for your attention. And this is what I tell my students all the time on a daily basis. Let's go do great things. And so I think that's true for maybe all of us. So thank you so much for having me, and I'd be happy to field any questions.
Great, thanks, Kyle. Really appreciate it. Wonderful talk, incredible work. I'm definitely going to join you for Topgolf when I can. Absolutely.
Great.
So let me open it up. Any questions, folks? Students, anything there? You can raise your hand, type them into the chat.
I have a
whatever you
quick
want.
question while
Yeah.
people are typing. So I'm thinking about the JAX 2021 article. It's the slide right before your substrate selectivity.
Mhm.
I'm sorry, I didn't see slide. The
Um,
yes, sir, that's right. I think no, the slide right above that. Sorry, one more.
yeah.
47. What's the difference in these evolved enzymes for the two that have the different in antiomeric ratios?
You mean the one where they flip?
Yeah.
Yeah. So it's been really interesting. So we actually I actually forgot to. I'm so happy that you just brought that up. I should have mentioned that we do get complementary selectivity for those, which is cool. The difference that we found, and one thing that's been tough to evolve actually for the Heister group in general, is when we were working on sort of the intramolecular one, there's sort of a hydrophobic pocket that phenyl group sits in, and that pocket is actually on the opposite side of the flavin in gluer versus no stock ER. I should mention, right, both of these were, okay, so no stock is definitely a wild type, but gluer was a wild type. We actually accidentally made the T36A mutation. At one point, we were going to try and mutate it out. We went ahead and screened that one, and it's been really, really effective. It turns out, and this is sort of one of the phenomenons about sort of directed evolution in general, is you'd actually think that would be in the active site. This is actually on the surface of the protein. It's crazy. So what we find actually is that I think it actually helps sort of stabilize one of the sort of the loops on sort of the outside of the protein and keeps it a little more stable. It doesn't degrade until I think 65 degrees, and the other ones degrade right around 30.
But then how do you prevent diffusion of your substrate and under those conditions, or are you highly saturating?
Oh, it's highly saturated. Yeah, definitely highly saturated. That's a good question.
Well, are you able to pressure like not just the promiscuity, but say on-off reactions through the light? And can you use different you know, so you're using like cyan light sources there too, right? But if you change the energy of the light, what can you do with that in terms of dictating towards one reaction versus another?
Yeah, so what we find, a lot of the changes that we found has been largely wavelength dependent more than it is sort of intensity dependent.
Okay.
So for example, like when we hit these, a lot of the thought process is not only does flavin sort of absorb sort of in the blue, but it also has a large sort of absorption band in the purple. But the problem is when we go to purple, like at 300 to 390-ish, we end up sort of sometimes photo exciting our substrate. So that ends up being a challenge that actually just falls apart, especially with these sort of alkyl halides. Intensity, though, I don't know if this directly answers the question, but these have required very, very intense sort of light outputs, right? So these are like pretty intense. So those, yeah, you probably saw in that one picture.
Yeah,
right?
the reaction chamber looks super intense.
Yeah, it was pretty crazy. We did do some work actually with Louis Campos at Columbia, where we're starting to work using sort of upconversion nanoparticles to do this. They're sort of aqueous nanoparticles, and we can start to do this now with red light, and it upconverts to I think to blue. We haven't optimized it for cyan quite yet, but yeah, I would say that's probably the most commentary I'd have sort of in terms of the intensity component.
It looks like we have a question kind of similar to that from Maria in the chat. I don't know if you can see that there.
Yeah, actually, I think should I come out of share, or do you want to read it?
I can read it. For the catalyzed reductive dehalogenation project, did you look at other wavelengths of light or just the 500?
Oh yeah, for sure. We actually built a little like a little machine that actually changes the wavelength and keeps it at the same intensity. So we scan anywhere from 300 and we go up by 5 nanometers each time. I don't know. I think up by 10 nanometers each time. We go anywhere from 300 to 650.
Okay. Yeah. Yeah. I had a question. Something about just at the very beginning too that you mentioned about the complexity of the drug candidates increasing.
Mhm.
Is that a result of resistance? What does that do to? Why are we seeing this continual increase?
Yeah, so that's a good question. I mean, I think so. I have two rationales for that. I think one of them is that we're starting to go after more specific targets. Even going after, instead of just enzymes, for example, metabolic enzymes, we go after sort of isoenzymes, even of the same type. The other thing I would say is, and maybe I should emphasize this more in talks, but I actually think we're just starting to get to the point where it's easier for synthetic chemists actually to build those molecules. Stereo complexity is still a major challenge right now, and so a lot of people are actually working on sort of taking those really complex molecules and doing selective transformations on those, right? So you don't have to go through a full resynthesis. So, but I would say still building sort of, in particular, stereo complexity is still a major challenge.
And I have a kind of a similar question on this from like just the whole idea, like in terms of the photobiocatalysis, et cetera. You obviously are gaining things from a green chemistry standpoint, but what is the, at the same point, are you fighting an uphill battle against going after more complex types of structures, or do you gain something in that direction as well?
Yeah.
Um,
So what's pretty cool, is as what we started to find, and this is something that I think people maybe don't appreciate so much in directed evolution, is as we've evolved these enzymes. So if you take them from a native function and you have just a little bit of overlap with new function, and then you evolve it for new function, those ones with new function are actually more catalytically promiscuous also. So they not only tolerate, so you usually get what you screen for, but what we started to do, as I was leaving actually Todd's group, is we'd actually start to explore those evolved enzymes that got really good sort of selectivity in this type of transformation for new types of reactions. And sometimes we're actually able to, one of the projects he'll put out eventually is that we're actually able to evolve the enzyme such that we can evolve away from cyan being the place that we need to hit it with light. So before we left, I think we evolved it from, I think 490 was its core sort of wavelength that we hit it at, to somewhere around, I think, 520. So we're getting pretty, it's moving a little bit. That's pretty. It doesn't seem that significant, but it's at least a little bit of a movement, and hopefully we can guide it even into the red and things of that nature. So, but I would say when we really need to have it work on something that's more complex, oftentimes what you'll do is sort of a substrate walk. So you'll start with something that's structurally similar, and then you'll start to kind of build sort of that complexity in until you get to that compound.
No, I'm glad you brought that up because that kind of reminded me of another question. Because when you were talking about that, it reminded me of just the entire when we shifted to this whole system robustness, right?
Yeah.
And when you're in that sweet spot, I was thinking, oh, you're building in robustness at this overlap area, but there's got to be some interesting potential in just driving it completely to this new catalytic state because you're inherently going to drive some new type of functionality and promiscuity over there as well.
For sure.
Yeah.
No, that's completely accurate. That's completely.
But it's really interesting from that perspective because you're almost building in this tool set of being able to dial in your robustness depending on what you're trying to hit. And
Yes.
and I assume like maybe even long term you could think that there's a potential to even act across different types of conditions. I know these are mostly solvent based, but if you're going towards some of the biocatalytic stuff, changing just the conditions there and how they function.
That's right. That's right. I know we started to even look at sort of that. I mentioned it briefly, but we're starting to even sort of the optimize around. We have sort of I don't know. There's a couple of people that say we can get some information on this, but we actually started to evolve even enzymes now under pressure. I'm actually finding that actually when we put them under just a little bit of pressure, they actually they have way different selectivities, right? Changing their conformation a little bit. That's really cool. So we can actually start to get sort of snapshots. Now I'm working on this a little bit with Petra and you can start to get snapshots of what that conformation looks like, and then start to evolve towards that. I think that's going to be a really exciting area to get
That's
after.
really- Jen, I see you have a question, and then we'll probably for time after that. Go for
Okay,
it.
yeah, no, actually, that just reminded me, and I won't go into it, but we're very interested in pressure in general for modifying activities. But that reminded me of sort of a niche field in biophysics where, a number of people, parts of our group, were trying to modulate an enzyme or a protein structure by hitting it with a specific wavelength of light, in particular, some terahertz, some other areas. And so that's very similar to the pressure idea, which is just bringing up that's awesome. I have a quick question just about the state of directed evolution. One of your slides was showing the screening of reactivity in just a microplate, micro well plate, or whatever you call it. And I know, maybe 7 years ago, people switched to, or some people were thinking about using like droplets and microemulsions and high throughput screening. And then of course there's a whole crew that was looking at hacking the MySeq to screen for these kind of things. What's the latest?
Yeah, the latest, I think, I check in, so we have at least a little bit of insight from the folks at Merck. I was just talking to them a little bit. The latest right now, it looks like they're turning mostly to sort of microfluidics in order to sort of start to optimize these. It ends up being one thing that I think a lot of people don't appreciate actually is that being able to do sort of microfluidics and things of that nature is much easier for enzymes they're using to perform a native function. So our enantios, or sorry, our enzyme loadings, even at 1 mole%, are really, really high in comparison to what they would actually be able to, in order to get sort of the readout, right? They need to be exceptionally low to be able to do it sort of with microfluidics. So there's still a pretty big barrier to sort of people, I guess, in industry right now, even being able to use them for sort of new functions, even the ones like P450s, where you hear a ton about those. But even those, I don't think have broken sort of the barrier that they need to quite yet. And the other thing actually that goes sort of the green comment that Gabe had is that one of the large sort of waste generation streams, especially on scale, is actually surprisingly in the water that people need to get rid of once it's sort of contaminated. And so one of the challenges, and I think one of the ones that we definitely want to work on, is being able to run these at sort of higher sort of concentrations. I think that's going to be one of the core sort of optimization areas right now. These reactions are run pretty dilute in comparison to what you'd normally run a typical organic reaction in. But progress is being made, but I think we're still sort of in the microfluidics area. A lot of people have actually tried to tie it to life. So actually it's like actual cell life, right? So you can actually optimize that way. But we haven't found a ton of good mechanisms to do that quite yet. I think that'd be a really exciting area to work in as well.
Awesome. Well, I'm going to, due to time, I'm going to call it there, but thanks so much, Kyle. Hey, do me a favor. I'll send you an email, but send me that retrospective. I really want to read that, and congrats on that. That sounds like a really important piece of work. And I know we would definitely like to get that out to our folks here at APMS and META. So, and through NCI as well. I think it's really important for students and faculty alike to see that. So congrats to you and your wife on that. And we'll have to have her give a seminar soon too.
That's right. Yeah. She'd love to.
Absolutely. All righty. Thanks, everybody. Thanks again, Kyle.
Thanks, Kyle. See you on campus soon, hopefully.
Thanks. See you soon, Trevor.
Yeah. Bye.
Yeah.
Thanks so much, guys.
Yeah, that was great, Kyle. Really, really loved that. Just tremendous work. It was really neat seeing it from all the different perspectives. I right when you put that slide up there, I was like, oh yeah, you got to be doing this work with Greg. I didn't realize he was your co-advisor back at Princeton.
Yeah, absolutely. Yeah, I developed really good ties with. He was a great person to work around.
Yeah,
really fun guy.
Are you still doing any work with them out there at Princeton with Heister and?
Yeah, so we have a little bit of collaboration. So there's another use that we have sort of for those some of the nanomaterials that we're using for up conversion between us, the folks at Columbia and them. But it's yeah, that's sort of where we've been kind of sitting in for now. I just talked to Greg actually when he was here visiting, I think 2 weeks ago or so,
Oh,
and he's got sort of an idea that he wants to collaborate on now. So